I welcome you all to the Global GOACON 2025 and extend my heartiest regards to all my seniors, faculties, juniors, friends, colleagues and students. Today I will be discussing upon the topic biopharmaceuticals and gene therapy. First of all, let's get to know the definition and the basics. Biopharmaceuticals include proteins and antibodies and oligonucleotides. They are used as drugs. They can be of two types, first generation and second generation. The first generation biopharmaceuticals are mainly the copies of endogenous proteins and antibodies, and the method of their production is recombinant DNA technology. The second generation biopharmaceuticals, on the other hand, they are engineered to enhance and improve upon the performance of the protein, antibody or antisense agent. Some well-known applications of the biopharmaceuticals are therapeutic monoclonal antibodies, recombinant hormones, a wonderful control of the gene expression, that is oligonucleotides. Now I would like to give a brief overview of the gene therapy. Gene therapy is nothing but it is the addition of genetic material to the cells to prevent, diminish or cure a disease. And now you can very well understand the applications of gene therapy. First is the radical cure of monogenic diseases like cystic fibrosis, hemoglobinopathies, and reduction in the disease with or without a genetic component, and that includes examples like many malignant neurodegenerative diseases and also infectious diseases. From very olden times, the proteins were used as therapeutic agents. If you remember the extraction of insulin from animal pancreatic tissue, extraction of the human growth hormone from the cadaver pituitary gland, these were the initial proteins that were used therapeutically, but there was always a problem arising from the administration of animal proteins to the humans. It was visible when there was a proper case of Creutzfeldt-Jakob disease due to the administration of human growth hormone derived from the cadaver pituitary glands. To deal with all such kind of problems, there has been a great improvement, enhancement in the technology with the advent of genetic engineering techniques. Now let's talk about protein and polypeptides. As I told you, the first generation and the second generation biopharmaceuticals are there. The first generation, they are produced by the recombinant DNA technology. One of the greatest examples and the first and the initial examples is recombinant human insulin that was produced in 1982. While the second generation biopharmaceuticals, they are engineered. There is a proper alteration of the gene so that the structure of the recombinant protein is actually transformed, and that is done deliberately to improve the protein's activity profile. Recently, among all of the production methods, the use of plants for the generation of the recombinant proteins has attracted a lot of interest. The human genes can be readily transfected into the plant genes, and that readily offers a lot of advantages. Examples are the use of lettuce and bananas to produce orally active proteins, such as vaccines, and they can be